Hello everyone, this video will talk about the complement system. The term complement refers to a complex set of distinct serum proteins present in normal human serum. The term complement refers to the ability of these proteins to complement, supplement, or to augment the different effects of other components of the immune system. They aid in the antimicrobial effects of antibodies. They are also considered to be an important component of the innate immune system. These are a set of complex proteins of approximately 20 in number. They are involved in 3 separate pathways of activation. These complement components are synthesized in the liver except for C1. These components are numbered from C1 to C9, and all 9 components consort or interact with one another. Fragments of these components are given the lowercase letters. For example, a fragment of the C3 component is named as C3A. A is usually given to the smaller fragment, while B is given to the bigger fragment. The complement system has 3 main effects, which will be discussed in the video. The first one is the lysis of cells, or cytolysis. Second one is the generation of different mediators. And the third one is opsonization. The components of the complement system involves more than 25 molecules whose biologic activities are numerous and closely related. 5 proteins are uniquely seen for the classical pathway. 3 proteins are unique in the alternative pathway, and 6 proteins are common in both pathways. These make up about 14 basic and distinct proteins. So what makes them more than 25? We need to add the serum proteins that modulate complement-derived activities, like the C1 inhibitor. Plus, we need to add other molecules that participate in the complement system, like plasmine. All of this would make more than 25 molecules that participate in the complement system. There are 3 pathways involved in the activation of the complement system, and these are the classical pathway, alternative pathway, and the lectin pathway. The activation of the different complement is often referred to as a cascade, which begins when one molecule is activated by certain reactions. And the 3 different pathways all involve the cascade of the different components. This is a table that differentiates the pathways according to their activation, the different requirements, and the unique components present in each. For the activation, the classical pathway needs immune complexes, or the antigen and antibody complex. The alternative pathway and the lectin pathway are both antibody independent, which means they do not need the presence of the antibody to be activated. For the requirements, the classical pathway needs at least 1 IgM antibody or 2 IgG antibodies, and we will discuss this later on. The alternative pathway needs microbial surface, or the cell walls of the organism. The bacterial cell walls have the polysaccharides or the lipopolysaccharides that are needed for the activation of the alternative pathway. The alternative pathway is more important the first time that an infection happens, because the antibody required to trigger the classical pathway is not yet present. The lectin pathway, on the other hand, requires the carbohydrates on microbial surface. The different unique components for the pathways are: the classical pathway needs C1q, C1r, C1s, C4, and C2. The alternative pathway has factor B, factor D, and properdin, while the lectin pathway has the MBL, or the mannose binding lectin. This is the cascade that is followed by the classical pathway. It starts with the presence of an antibody which is bound to an antigen. This is the activation of the classical pathway. The first component recognizes this presence of an antigen and an antibody and starts the activation of the different components of C4, C2, C3, and C5. C5 forms the first membrane attack unit, and this further activates the different components from C6 to C9, which makes up the membrane attack complex. Let us now discuss the classical pathway in detail. The activation of the classical pathway starts with the presence of the antigen and the antibody complex. The first component of the classical pathway is the C1. This is the recognition unit of the classical pathway, which is a macromolecule held by calcium. It is a trimolecular complex with 3 subunits of C1 called C1q, C1r, and C1s. C1 appears as 6 globes held together on slender shafts that fuse into a common base. It is also considered as a pentamolecule because it consists of a single C1q, 2 C1r, and 2 C1s molecules. Remains of the C1 molecule binds to the second heavy chain portion of an IgM or 2 IgGs. The globular domains are the recognition unit that bind to the Fc region of the immunoglobulins. C1 needs to bind to 2 Fc portions. That is why 2 IgGs are needed. Since IgM is a pentamer, the presence of 1 IgM will initiate the cascade. Once the C1 complex is attached, this leads to a conformational change in the C1 complex that leads to C1s molecules to give active serine esterases. This starts the third stage, which is the activation of the C4 component. C1s activates the next component, C4, and is cleaved to C4a and C4b. C4a has no further part in the complement sequence. C4b attaches to the microbial surface. It does not attach to the antigen-antibody complex. C4b now acts on the binding site of the second activation unit, which is C2. When combined to C4b, C2 becomes a substrate for C1s and is cleaved into C2a and C2b. C2b is very labile and will decay very fast. C2a remains bound to C4b in the presence of magnesium to form C4b2a, known as C3 convertase. C3 convertase is called as such because it splits C3, which is the most abundant complement component. It splits C3 into C3a and C3b. C3a is released into the plasma, while C3b combines with C4b2a to form C4b2a3b, also known as C5 convertase. This is the most significant step in the complement activation series. C5 convertase cleaves C5 into 2 subunits, C5a and C5b. C5a is released into the body fluids. The generation of C5b starts the formation of MAC, or the membrane attack complex. C5b binds with C6, forming C5b6. And this then binds to C7 to form a C5b67 complex. C8 and C9 then sequentially bind to this complex. C9 forms a pore-forming molecule containing up to 14 C9 monomers. This results in the formation of the lytic plug. This is the first function of the complement system, cytolysis. Lysis is brought about by the entry of water and electrolytes into the cell through the disrupted cell membrane. This cascade happens simultaneously to different parts of the cell and to all antibody-bound cells, including bacteria, tumor cells, and erythrocytes. The second function of the classical pathway is the generation of mediators. C4a, C3a, and C5a that were produced during the classical pathway activation are all considered as anaphylatoxins. These components act on mast cells and basophils for degranulation. These cells will then release mediators like histamine to increase the vascular permeability of the blood vessels. This allows an increased leakage of other complement and other plasma proteins to the site of infection. Among the three components, C5a is the most potent. It is also considered as a chemotaxin or a chemoattractant that leads phagocytes like neutrophils and macrophages to the site of activation. The third function of the classical pathway involves opsonization. C3bs are opsonins. Their presence on the surface of the cells makes phagocytosis better and more efficient. The second pathway is the alternative pathway. The microbial cell wall activates the C3 conversion, leading to the binding and splitting of factor B. C3 convertase is formed and properdin is bound, cleaving more C3 and leading to the C5 convertase. This eventually forms the membrane attack complex, which is similar to the classical pathway. The alternative pathway is antibody independent. It does not need the presence of antibodies to activate this pathway. The alternative pathway is more important the first time of infection by microorganisms because that antibody required to trigger the classical pathway is absent. All it needs is the cell surfaces that are foreign to oneself, like bacterial lipopolysaccharides or endotoxins, fungal cell walls, or viral envelopes. So how does the alternative pathway work? One thing to know about the protein C3 is that it spontaneously is converted into C3a and C3b. It does not need to be activated to be converted. There is a continual hydrolysis of C3 in the plasma. Normal cells, normal healthy cells, can inhibit the effect of C3b. Once C3b attaches to the cell walls of the normal cells, these normal cells can deactivate the effect of C3b. But foreign cells like bacteria, viruses, and fungi do not have the inhibitory protein to deactivate C3b. If not deactivated, this then starts the alternative pathway. The C3b that is attached to the microbial surface then acts as the binding site for factor B. This is the first unique component of the alternative pathway. The second unique component is factor D. Factor D cleaves factor B, which is bound to C3b to form BA and BB. The fragment BA is released while BB stays with C3b. The C3b-BB is the C3 convertase enzyme. So again, the C3b-BB is the C3 convertase of the alternative pathway. Properdin, which is the third unique component for this pathway, binds to this complex to stabilize it. The stable C3 convertase then cleaves further C3 into C3a and C3b. C3a is released, while the formed C3b binds to the C3 convertase to become C3b, BB, C3b complex, known as the C5 convertase. This continues to the final phase of activation of the complement cascade, which is similar to the classical pathway. C5 convertase cleaves C5 to C5a and C5b. C5b binds to C6, 7, 8, and 9, and consequently forming the lytic plug. The third pathway is the lectin pathway, also known as the Mannan-binding lectin pathway, or MBL. This pathway does not require an antibody for activation as well. Activation is brought about by the mannose-binding or Mannan-binding lectin. Lectins are proteins that can bind to carbohydrates. MBL is produced in the liver and is normally present in serum. MBL is also an acute phase protein, therefore increasing during inflammation. The lectin pathway requires the activation of MBL, or mannose binding lectin, which is a unique component of the lectin pathway. The word mannan refers to the polymer of mannose. MBL is somewhat similar to the C1 component in structure and function. C1, as mentioned earlier, binds to the Fc portion of IgGs or IgM. A lectin is any protein that binds with sugars or carbohydrates like mannose and glucose, which are found on the surface of microorganisms. MBL is associated with other proteins called MASPs. MASP stands for MBL-associated serine proteases. The MASPs are very similar to C1R and C1S molecules of the classical pathway in that it cleaves C4 and C2. So this is how the lectin pathway works. The carbohydrates found on the surfaces of the microorganisms would be recognized by the MBL, and the MBL will attach to them. MBLs are normally found in serum, so when organisms are seen, they would be recognized by the MBL. Once the MBL is attached to the carbohydrates like the mannose or the glucose on the microbial surface, the MASP2 will then act like the C1, where when C4 is attached to it, it will cleave it into C4a and C4b. It then follows the same pathway as the classical pathway. C4a is released while C4b is bound to the cell surface. C4b is then attached to C2a forming the C3 convertase and attached to C3b forming the C5 convertase. It then follows the same pathway to form the MAC, or the membrane attack complex, until lysis is formed. To summarize the different actions of the complement system, first is that it increases the vascular permeability done by the components C3a, C5a, and C4a that have been released to the body fluids. These components also cause the degranulation of mast cells and basophils that also leads to the smooth muscle contraction. C5a acts as a chemotaxin for phagocytes like neutrophils and macrophages, while C3b acts as opsonins for phagocytosis. And lastly, complement system ends in the lysis of bacteria and foreign cells with the formation of the membrane attack complex. Let's now give a recap on the three major independent yet overlapping pathways for complement activation. In the classical pathway, immune complexes bind to C1, cleaving C4 and C2. The fragments C4b and C2a form the C3 convertase, which is C4b2a. The C3 convertase cleaves C3 into C3a and C3b, and the binding of C3b to the C3 convertase forms the C5 convertase composed of C4b, 2a, and 3b. The lectin pathway binds sugars on microbial surfaces. MASPs take the place of C1 proteases. The alternative pathway continuously generates C3b that deposits on pathogens or debris. C3b engages factors B and factors D to form a C3 convertase C3b-BB. The binding of another C3b to the C3 convertase generates the C5 convertase C3b-BB-C3b. The succeeding cascade is common on all three pathways. C5 is cleaved to C5a and C5b. C5b engages the terminal pathway and initiates the formation of the lytic membrane attack complex. The end result is the lysis or the rupture of the cell. To emphasize the importance of the complement system, let's give some examples of conditions that may occur if a deficiency of the components occur. A deficiency of the C1QRS have been described in patients with SLE-like symptoms and increased susceptibility to bacterial infections. Decreased C4 levels, accompanied by elevated anti-DNA and ANA titers, confirm diagnosis of SLE, also associated with glomerulonephritis and pyogenic infections. C2 deficiency is the most common of the human complement deficiencies. Patients with C2 deficiencies have chronic renal disease, also associated with autoimmune diseases and are susceptible to bacterial infections. MASPs deficiency are prone to pneumococcal disease. Properdin and factor D are associated with pyogenic and Neisseria infections. A deficiency of C3 leads to severe, recurrent pyogenic and respiratory infections. Included herewith is glomerulonephritis. C5 dysfunction, also known as Lehner's disease, are predisposed to skin and bowel infections characterized by edema. This is also seen in patients with SLE and are prone to Neisseria infections. C6 appears to be associated with increased susceptibility to Neisseria infections as well as SLE, Raynaud's phenomenon, scleroderma-like symptoms, and vasculitis. C7, 8, and 9 are associated with severe bacterial infections caused by Neisseria species. And that ends this complement system video. Thank you very much for watching.